So thank you for everyone who's joined us today, who's taken some time out of their day to join this really exciting webinar, Go With Your Gut, where we'll be sharing some novel findings on the gut microbiome, nutrition and health. So as you'll see, I'm joined by three really wonderful scientists today, who are all authors on a new paper that was published in Nature Medicine today. So Nicola, Sarah and Tim, could you each introduce yourselves, just give our viewers a bit of background into where you're from and what you do. Nicola.
Hi everyone, thanks so much for joining. So I'm Nicola Segata, I'm professor at the University of Trento, Northern Italy. And we work on studying the gut microbiome, and this I think is a very exciting analysis that we did on the links with diet and cardiometabolic health.
Great. Well, it's great to have you here. And Sarah, I know you've joined our webinars before. Would you like to just introduce yourself to our viewers?
Yeah, thanks, Kirsten. So I'm a nutritional scientist at King's College London, and my main area of research interest is to do with diet and cardiometabolic disease, postprandial metabolism, and personalized precision nutrition. And I have extensive experience in the running and setting up of large randomized controlled trials.
Great, thanks, Sarah. And Professor Tim Spector, it's lovely to have you here. Can you just give a bit of an introduction for those who don't know you yet?
Yeah, so it's Tim Spector. I'm Professor of Genetic Epidemiology at King's, and I am medically trained in epidemiology. I worked with twin studies, worked with the COVID symptom study app, set that up in the UK and the US. And the last 10 years I've had an interest in the microbiome and gut health, and I've written a couple of books on nutrition, The Diet Myth and Spoon-Fed.
Great. Well, it's lovely to have you all here. So just as a bit of an introduction, I guess, before we get started, some housekeeping. So we are live streaming on YouTube, so please feel free to ask questions in the comments there. On Zoom, you can ask questions in the Q&A section here, and we'll try and answer as many of them as possible as we move through the webinar. So a bit of an introduction to today. I am Kirsten and I work at Zoe. I'm a registered associate nutritionist, and we've basically decided to put on this webinar today because we have some really exciting new research that's been published in the leading journal Nature Medicine today. So a central part of our research at Zoe has been the investigation of the gut microbiome and its links with diet and metabolic health. So today we're going to give you a bit of an insight into what this research found and what this means for you. So Tim, as I know you're a gut microbiome expert, it would be wonderful if you could just give our viewers a bit of an insight into what exactly the gut microbiome is and why we should care about it.
Well, everyone needs to know about it because essentially it's like just in the last 10 years, we've discovered a new organ in our bodies. And this contains hundreds of trillions of microorganisms, including bacteria, viruses, and fungi and parasites, all living together in our gut from our mouth down to our bottoms, most of them in the lower colon, the lower part of the intestines. And this is a very complex ecosystem of microbes. And of course, each of these microbes have their own genes. So they have hundreds of times more genes than our own human bodies have. This is important because they all produce chemical proteins, and they're essentially chemical factories. And this has a huge general role across our bodies, not only in creating these metabolites and enzymes, many of which are unique and can't be produced by our own cells. They produce vitamins. They're important for our metabolism, our appetite, our immunity, and that's particularly important at the moment with COVID, and much more. And you can read about this through various overviews, such as this one in the BMJ that we wrote with Ana Valdez. So it interacts with every medication you're taking as well. So it's a two-way process. So the microbes affect us, and anything we do to them, including food and chemicals, affects them. So it's a very complex process, but really crucial to understanding the nutrition in a new way and our health in a new way.
Great. So yeah, exactly. So the gut microbiome is associated, we know from kind of a large body of research, with things like heart disease, type 2 diabetes, inflammatory bowel disease, obesity and depression. And kind of the research as a whole generally associates kind of a higher gut microbiome diversity with better health. Now, Nicola, I was wondering if you could just give our viewers a bit of insight into what gut microbiome diversity is and what we've also learned about it through our research.
Yes, so with gut microbiome diversity, we refer to basically the amount of different distinct microbes that are populating our gut. And this is important, no? Because an healthy ecosystem usually means an ecosystem with many different players. So the same is with the microbiome. So usually a high diversity, now many different bacteria living together in our gut usually means unhealthy microbiome. And this has been known for a bit of time, and we confirmed it also here in our study. However, what I think is important is that although we look also beyond just the diversity, and we will see that we'll find a lot more and stronger links than just with general microbiome diversity.
Great. So, you know, until now, we know that, as Tim mentioned, the bugs in our gut are associated with our food. They're influenced by the food we eat. They impact our health. But until now, it's been really challenging to actually unravel all of these complex links. And as we'll kind of see soon, the research we've been doing at Zoe has actually allowed us to look deeper than has ever been kind of looked before. So we know that humans share around 99% of the same DNA, but we actually share on average less than 30% of the same gut microbe species with each other. So, I mean, what does this mean, Tim and Nicola, if you could just comment a bit on this, like why is this important and what does this mean for us?
Well, I think we always assume that any differences between us in our response to something like food was down to our genetic differences. But and there certainly are genetic differences between us, but because we're actually so related, we're on average fifth cousins. There's actually less difference between us than perhaps we imagined. And so the difference between the genes in our microbes is very much more than anything we get from our DNA. And that's why, in a way, looking at your gut microbes is a much better reflection of your general health than looking at your DNA, because of this huge range and the ability for it to vary. And we look simply at identical twins who share the same species. And Nicola, I think, did some more detailed analysis looking at the strain level. This is the subspecies level. I found some really interesting facts there, Nicola.
Yes. It's so we can look at our own genome, no? And our own genome, we know, is very similar between person and person, although each of us is unique also at the genetic level. We can also look with the latest tools that we are available also in the genetics of the microbes present in our gut. And so even though we see here unrelated persons, so for example, two individuals following us today in this presentation will share on average 30% of the species in their gut. If you take the species that are in common, for example, probably is very likely that two persons in this presentation will have Escherichia coli in their gut. If we look at the genetic variants of the Escherichia coli, we will probably never find the same exact variants into unrelated persons. So this means basically that at the microbiome level, we are even more unique than at the human genetic level.
Yeah, that's amazing. So basically, no two people have the same gut microbiome composition. So another really key thing to mention about the gut microbiome is the fact that we have some control over it and it can change. So there's a large body of research that shows the impact of diet on the gut microbiome. Now, Sarah, as a nutritional scientist, I was wondering if you could just comment a bit on this and give a few examples of how the foods we eat might impact the gut microbiome from the research we've seen already?
Yeah, so just as we've seen with some of our early analysis, that how we respond to foods isn't determined by our genetics. As Tim and Nicola said, our microbiome isn't also determined by genetics. And so I think this is really important because it's quite empowering. It shows us we do have the power and the control to change not only how we respond to food, but also our microbiome that will also impact this as well. And as you said, there's a growing body of evidence around how what we eat will impact our microbiome. So there's evidence around dietary fibers, for example, and different types of dietary fibers, soluble, insoluble, polyphenols, fats, etc. And what some of this research shows is just how variable our microbiome response is in relation to these foods. So if we take polyphenols, for example, which are found in polyphenol-rich foods such as berries, tea, cocoa, for example, how one person responds to it in terms of how it modifies their microbiome versus another is hugely variable. But what's really important is that we need to remember that our diets are really complex, and we need to think not just about the research that's been published around some of these single nutrients or single foods, because we don't consume single nutrients, we don't consume single foods. We eat meals as part of this wider dietary pattern, and this adds a whole layer of complexity around nutrient-nutrient interactions, around the food matrix impact. And given that our microbiome is really complex, if we overlay the complexity of food together with the complexity of us and our own unique biology, it's really difficult to actually disentangle these associations between diet, between food, between dietary patterns, the microbiome and health outcomes. And it's just not really been possible before, because you need such large scale studies, but also at a really high resolution. So high precision, high depth and high breadth. And this is what's really exciting about this research, that it's the first study that's of the adequate scale and depth to really be able to start to disentangle these associations across what we're eating, our microbiome, and also health outcomes as well.
Yeah, so one really key thing, I guess, to remember, the fact that each of us has a unique gut microbiome, which can be changed through diet, makes it a really exciting potential target to actually improve our microbiomes and health. So that's something to just keep in mind as we go through these findings, why they're actually important for us. So PREDICT is the world's largest in-depth program of research into diet and nutrition, and we've kind of been conducting this over the last 3 years. Sarah, would you just like to give a brief overview of exactly what we gathered in PREDICT and how this has been helpful in, I guess, allowing us to disentangle the relationships between the gut microbiome, food and health?
Yeah, so PREDICT is a whole program of research encompassing a number of studies, the first of which is the PREDICT-1 study that we're talking about today. And what makes it unique really is what you've already said. So the breadth of the study, the depth of the study, and also the scale of the study, and just the kind of resolution of data that we've been able to capture. It's a human metabolic study, and it involved a very tightly controlled clinic component as well as a free living at-home component. And what we've been able to do is, for the first time, study the gut microbiome and dietary contributors and health outcomes at the kind of scale and complexity that hasn't been done before, that, like I said before, we need to in order to unravel these different contributors. And we've been able to combine together very in-depth microbiome analysis using some of the really latest cutting-edge technologies and computational biology that we have experts such as Nicola leading on, and combining this with a very detailed dietary information that we've collected from everyone, and a whole breadth of health outcomes, body composition measures. And also quite uniquely, we've looked at postprandial responses. And when I use the term postprandial, I mean post-meal. So the changes that you have in some of these health measures and circulating metabolites straight after you've consumed a meal, which is actually really important because this underpins a lot of the long-term effects of what we eat or on health outcomes. And so we've been able to really gain these unprecedented level of insights into this hidden world of the microbiome, this kind of black box. And, you know, we've got therefore this largest set of metagenomic sequenced gut microbiome data that we can link to this diet and these health outcomes.
So I have a question. You mentioned metagenomic kind of data. What exactly is that, and what makes it so important?
So I think that's definitely one for Nicola to answer.
Yeah, so metagenomics is a relatively novel technique that really allows us to look inside the microbiome at very high depth. Basically, it's just the sequencing, so reading the genetic material of a whole microbial community. So you take 1 drop of saliva for the oral microbiome or 1 gram of feces for the stool microbiome. And basically, there are experimental techniques that are able to free the DNA out of the cells present in these samples. And then using high-throughput sequencing machines, basically the same machines that are able to now read very well the human genome, we read the little pieces of the DNA of the microbes. And we read the microbes, the DNA of the microbes of the whole community. So then is a computational, it's a computer effort to try to reconstruct the genomes, because it's already difficult to reconstruct the human genome. But in a metagenomic sample, we have genomes from multiple bacteria and microbes all together. So it's like trying to solve many puzzles together that are very difficult, because there are really a lot of readings of the DNA. So metagenomics, again, is just the analysis of microbial communities using sequencing and without trying to cultivate each single microbe present in the community.
Great.
Yeah, and just to add to that, Kirsten, because a
Yeah.
lot of people might have heard of microbiome testing with the 16S, and most of the data in the last 10 years has been with using this 16S microbiome sequencing, which is different to metagenome sequencing. So that method just looked at one gene. So it was very common, every microbe had it, and it was called the 16S gene. So that was a very crude way. It's a bit like studying people by looking at their blood groups and working out our HLA types. So we've advanced a lot. It was useful and it was much cheaper and quicker, but now we've moved on to this new way of actually sequencing every gene in every microbe rather than just one.
Yeah, that's a really good explanation. Thanks for that. So kind of with this depth of data, especially into the gut microbiome using these techniques, in our paper, there were a few key findings that kind of came out. So Tim, would you mind just taking us through what these key findings were?
Yeah, I mean, there's a huge amount of data in this paper, and it's hard to know what to pick the best bits are. But I think that the take-home message is really this, that we found a clear panel of microbes that were either positively beneficial markers of health or negatively associated with health. And for the first times, we linked this up with specific foods and individual microbes. So we've got some examples of that coming up, but basically it was the idea that we linked the foods and the microbes, and then found these strong associations between the microbes and these dietary patterns, both healthy and unhealthy.
Yeah.
And really we put this together. So we identified some very particular microbes that hadn't been looked at before, often without names, ended up having the strongest links to both the diet on the one hand and to our health, particularly involving our heart or our metabolism on the other. So it was the key link between diet, microbes and health that hadn't been done before that was really important here. And we'll go into a bit more detail on that.
Yeah, so hopefully we'll be able to break these down into kind of, very data heavy paper, into something that all of us can understand and relate to. So we're going to use kind of the terms good and bad microbes throughout this presentation. And Nicola, in our kind of session the other day, you explained to me what we mean by good and bad bugs. Would you mind just kind of explaining to our viewers what we mean when we use these terms.
Yes, so these terms are pretty informal, if you like, but we use these to refer to these panels of microbes that we were quite surprised actually to identify, because these are microbes at the same time, their presence in abundance is associated at the same time with healthy diets, so diets that are healthy from multiple viewpoints, if you like, but also at the same time, again, with favorable cardiometabolic markers, both at fasting conditions, so in normal conditions, let's say, but also postprandially, so the spikes that we may have in these markers after eating a meal, but also at the same time with obesity markers. So this means that, you know, at the same time, these bacteria are connected with all these aspects of human health and nutrition, which is highlighting the complex interconnections that are there. 15 are the most strongly associated, positively associated markers that are microbes, sorry, that there are more. And at the same time, there are bugs associated, microbes associated with in the opposite direction. But maybe it's easier to make some examples. So for example, Protella copri is a bacterium present in 30% of the population, more or less, at least the Western style population, let's say. And if you have it, that is associated with having an healthier blood sugar levels after eating, so postprandially. The same thing is for blastocystis, similar prevalence, present in 30% or so of the population. And this is a eukaryotic microbe, so a parasite, which has been very overlooked by other analyses and other studies. And if you have it, again, this is associated in the same time. Interestingly, if you have both Protella copri and blastocystis in your gut, the health association, the association with healthy markers is even stronger. Firmicutes bacterium CAG95, this strange name is what Tim was referring before. These are bacteria that have never been really cultivated. So we don't have a good name because nobody was able to characterize it in vitro by growing it and analyzing it. Nevertheless, we can find it through metagenomics, as I was saying, and these really uncharacterized and obscure, if you like, bacterium is really one of the top scoring in our panel of these 15 good, as we say, microbes. On the other hand, you have the bad guys. We mentioned a couple of them here, Flavonifractor plauti or Ruminococcus gnavus. Those are, they have the opposite association. So we tend to find it more in people with unhealthy diet and with markers of cardiometabolic health that are not very good. And at the same time, this is associated also with bad obesity markers like BMI or visceral fat.
Great. So for anyone watching who's maybe wondering, you know, what are healthy blood sugar levels? What are unhealthy blood fat levels after eating? After the webinar, the joinzoe.com/blog, there are loads of articles that go into detail on these kind of things, and I'll make sure to link to them in any show notes on the YouTube video. So one of the things we discovered, so we obviously discovered these links between microbes and health, but we also found very clear associations between gut microbes and the foods that we eat. So Sarah, would you mind going into a bit of detail about exactly what we found and why this is important and so novel?
Yeah, so I think Nicola and I'll follow up with some really specific examples in the next few slides, but to put into context the following slides is really important to point out that we looked at all the levels that we need to be looking at when we're doing nutritional research studies in terms of what we're eating. And so we took this kind of four-tiered approach where we looked at nutrients, we looked at single foods, we looked at food groups, so for example, dairy, red meat, for example, and we also looked at dietary patterns. And when we use the term dietary patterns, we're talking about the overall pattern of the foods that you're consuming. So for example, we know that a Mediterranean diet is associated with good health outcomes and also with a healthy microbiome. And in terms of nutritional research, the kind of landscape is really changing, like I said earlier on, to move away from looking at just single items and single nutrients. And this is what we've been able to do in the research and what will go on in the next few slides to show you, that we can kind of disentangle the relative importance of the nutrients versus the foods versus our overall dietary pattern. And this, from a kind of implementation point of view, is really important, because it means that we can actually give actionable advice that's relevant to you as a person. It's all very well saying this particular fatty acid is associated with this bug, but how do you translate that into your own dietary pattern and into modifying your own, individual food choices?
Yeah, so kind of Sarah mentioned there were a few specific examples we would pull out. So yeah, Nicola, I was wondering if you could just pull out some of these examples that we have on the screen. So things like coffee, discuss yogurt, and how obviously milk chocolate and dark chocolate we can see on very opposite ends of this heat map, where we can see these distinct clusters.
Yes, this is maybe a bit of a complex chart, if you like, but sometimes it's impossible really to simplify too much. Metagenomics also means big data, basically. So here, this heat map is just reporting some of the associations between specific bacteria and specific foods. It's only a small fraction of all of them. The point is that the darker the red, the stronger is the positive association, blue is vice versa. And as you were mentioning, there are some expected links, and one of them, you can spot it there probably, but I can tell you, is the link between Bifidobacterium animalis and yogurt. This is not surprising at all, because actually Bifidobacterium animalis is actually present and alive in most yogurts. So this was expected, and it's nice, of course, to see some expected associations to come out. But coffee, for example, was not expected, also because the association is again with uncharacterized bacteriums, a very unknown one. And that was one of the strongest associations. This has to do with biology. It has also to do with probably the fact that it's easy to survey how much coffee one eats. So coffee drinker and non-coffee drinker is a very black and white condition, probably. But for example, tea does not have the same effect. Tea that is not associated with the same bacterium with which coffee is strongly associated. Other little examples and that are pulled out here is the one of the chocolate. So in general, in this chart, you see two different colors. This means that the panel of bacteria we were mentioning before are really associated with different kinds of diet. And interestingly, dark chocolate is on the good side. So it is in the panel of the good bacteria associated with the healthy diet. Milk chocolate is on the other side. Of course, we don't want to overinterpret it. We know that dark chocolate is probably associated with healthier diet because of choices of persons eating better than others. But again, our work was really try to dig inside all these set of associations to find those that are the most relevant for us and for our unique biology.
Great. So as Sarah mentioned, we also looked at the relationships between microbes and food groups. So what was really interesting here is, I guess, not so much that we found that those with diets rich in healthy plant-based foods were associated with good gut bugs, but actually that these same kind of good gut bugs were seen in those with diets that contained healthy animal-based foods. So Sarah and Tim, kind of, why is this so surprising or interesting, and what does this mean for kind of how we approach food?
Tim, do you want me to start?
I'll start with you. Okay. Broadly, it means that we can't just rely on the old-fashioned categories of what's healthy and unhealthy, and you know, it's much more nuanced than that. And so the idea that that is around there, that anything that is plant-based is good and anything that's meat-based is bad, is really not true at all. And this really helps build on this idea that the way we look at food has been much too simplistic in the past, and that actually it's the individual composition of those foods, not this broad, more cultural definitions that are really important. And this is what we're seeing, that our microbe associations are cutting through these categorizations and showing that the stuff that's generally highly processed, whatever the origin of it, is tending to come out as unhealthy, and this seems to be working through it. So it's really just saying that food quality really matters, and that dietary patterns rather than just individual nutrients are also really important.
Yeah. And Sarah, do you have anything to add to that? Or I think Tim's kind of covered everything.
Yeah, I think he's covered it all. I think just to reiterate that his point about the food quality and about the level of processing is so key. And where we see the segregation between the healthy plant and unhealthy plant foods, we see the same segregation in the bugs, just in a similar way to how Nicola showed you that segregation in the slide before. And we developed a novel animal index where we separated animal foods according to whether they were healthy and unhealthy. And again, we saw that same segregation. So it's really about moving beyond, like Tim said, those very traditional classifications and thinking about the food quality, the level of processing, etc., when we're thinking about how good a food is for our microbiome and also for our health.
Yeah, so kind of finally, we've talked about these links between microbes and food that we discovered, and kind of now, so we saw this heat map earlier, and this is our attempt to kind of simplify that a bit into something that's really, it's a lot more understandable for me. This is kind of to try and illustrate how we also discovered these clusters of good and bad bugs were actually highly associated with markers of better health and worse health. So Tim, could you just dive into this in a little bit of detail, and just explain anything we haven't covered relating to these findings?
Yeah, so I think what other studies have done in the past is take some individual microbes in mice or rats and shown some changes to them, or in large case control studies, but generally they've not done multiple risk factors, and they haven't done this at any scale in humans. So what we've done is an unprecedented depth of looking at the health markers in thousands of individuals across countries to make sure that these associations are really robust. And this is what we're showing, is that basically our cluster of good bugs are associated with much better fat control. Means your fat levels don't go up after a meal as much. Your blood sugar control is less for that 30 minutes after eating. And this is related to lower levels of blood inflammation, lower levels of body fat measured by DEXA scans. This is the abdominal fat, etc. Your heart disease risk and your diabetes risk are going down. And the opposite is true with the so-called bad bugs that we have seen associated with poor food choices, etc. So I think this is what's different, and we've looked at this consistently. So Nicola actually checked a lot of these associations, particularly against obesity, and found they were consistent in nearly all the published data sets around. So we're pretty confident this isn't just a chance finding. We'd find these associations around the world.
So we've had a few questions actually about, what is the link between the gut microbe and type 2 diabetes? What is the link between the gut microbiome and heart disease risk? And how might these findings inform our understanding of that in future?
Well, up to date, really, people have just so far seen, for example, heart disease risk or diabetes risk is associated with lower diversity, less numbers of species, and a few microbes have here and now been implicated as being important. But most of them haven't replicated in many big data sets. So we're still sort of unclear about what's going on. What we're showing here is that it is complex. We can't blame one microbe for this. But at the same time, we are identifying is that microbes that are important are now also linked to diet. So if we can manipulate the diet, we can change a small amount of each microbe, which together would have a much more major effect in shifting our microbes, which would then shift our health. So we're moving away from this, everything's caused by one microbe and you have to get rid of that, to this more general consortium of microbes, which wants the green ones to have more of them and we want the red ones to have less of them. That's really what we're after. And we think that this may go beyond just what we've tested. So people are asking about depression and anxiety a lot, and we are seeing lots of overlap between microbes that are bad for inflammation with those that are bad for mental health as well. So, and studies have shown that you can improve mental health with good diets that affect your microbiome. So I think we're going to see much more of this. This is really the exciting tip of the iceberg. But I think this is the essential first step to be sure of what we're finding.
Yeah, that's amazing. So kind of, I guess lastly, and this might be something that people, those watching may find a lot more relevant just to their day-to-day lives, is, gut microbes actually impact our metabolism, so how our bodies process the foods that we eat. So Tim, would you like to talk a bit about how the gut microbe, well, I guess what we know about the impact of gut microbes on our responses to foods? And then Sarah, would you like to touch on why this actually matters for us?
So I'll start off. I mean, so to be honest, we don't know exactly how our gut microbes do this. They've just got all these amazing chemicals at their power. They can release, they can turn up, they can turn down. And because they can reproduce every 20 or 30 minutes, they can suddenly, turn these knobs up and down in response to food. And we think that's what they're doing. So that when they sense that certain food's coming, they will change the amount of chemicals in the body, which will either change the way our gut works or break down that food faster. So we know that they influence our response in that first couple of hours to a carbohydrate sugary meal. And some people will have a very good response and the sugar and insulin are kept under control, others won't. And the same is true with fat at 6 hours. We know that microbes, we know a bit more about how microbes affect fat, and that's through the gallbladder and release of bile salts. But also there are particular microbes that just love eating fat, and they digest it and they then produce other chemicals from it, which makes the body more or less able to get rid of that fat quickly from the rest of the body. And I think Sarah can explain why these peaks and getting rid of them is so important.
Thanks, Tim. I think to put it into a little bit of context in relation to the figure that you've got up here, Kirsten, would probably be helpful first. So the top figure here, we have the change in circulating blood sugar after consuming a carbohydrate-rich meal. So when any of us consume a mixed meal containing any kind of carbohydrates, you get a peak in sugar in your blood. Around 30 minutes, returns to baseline around 2 hours. And what you can see with the pink line here is quite a big peak. And a little dip, and with the blue line you see a smaller peak. Ideally what we want to do is keep this peak smaller, and I'll come on to explain why. And it's the size of these peaks, like Tim said, that we've seen to be associated with a particular cluster of microbes, and the same cluster of microbes that we've seen associated with these healthy diets and other health measures as well. And then on the figure below, this is the change in blood fat, so circulating triglycerides in the blood when you consume a meal that contains fat. And it has a slightly different pattern of response, that it peaks around 6 hours, turns to baseline around 8 hours. And again, you can see a differentiation between this blue and this pink line between what we consider a healthy blood fat response, which is the blue line, versus this more exaggerated unhealthy fat response. And again, these are influenced by the microbiome. And this is actually the first study ever to show that. So no one's ever looked before how the microbiome is associated with this change in circulating blood fat levels after consuming a meal. And the reason that we want to keep these peaks to a minimum and prevent throughout the day all of these kind of metabolic fluxes that you have after you've consumed a meal is because when you have this rise in either glucose or on fat or both of them, you have a whole host of knock-on effects on other circulating metabolites. So these are inflammatory mediators, oxidative stress, certain lipids circulating the blood that we know increase your risk of heart disease, as well as the impact from the dips in the sugar on hunger, satiety, subsequent energy intake. So there's a whole host of detrimental knock-on effects from these peaks. And if they're too high, we know that therefore they increase our risk of a whole host of metabolic diseases. And we now know that they're actually an independent risk factor for cardiovascular disease. And it's these acute changes that we know underpin many of the chronic effects, so many of the long-term effects that we see from the foods that we eat. So that's what's really novel again about this work, that we didn't just look at fasting values. As well as the whole breadth of fasting values, we specifically looked at these very acute responses to see how they relate to the microbiome, which I think is, particularly related to the fats, is really exciting data that we're showing in this paper.
Thank you for that. So once again, I've actually seen a few comments like, how do I read these charts? So we'll try and talk through this one and make a bit of sense of it. The original heat maps are even more confusing, but this chart is kind of meant to bring together all of these things we've been talking about. So through this research, we've identified these good bugs and bad bugs that kind of consistently were linked to markers of health and diet. So in the top left-hand corner, you can see these are the good bugs, which we identified were consistently linked to, good blood fat and blood sugar responses, less inflammation, lower levels of body fat, lower heart disease and type 2 diabetes risk. And similarly, these good bugs were linked to healthy dietary patterns, healthy plant-based foods. Your bad bugs in the bottom left-hand corner on the other side, these were negatively associated with the good markers of health. So they were associated with, poor blood fat and blood sugar responses after meals, more inflammation, higher levels of body fat. And so, Sarah, would you like to just touch on kind of anything I've missed in this diagram and maybe once again, just bringing it all together, kind of why this matters for us?
Yeah, so I think really just to reiterate that what we've really been able to disentangle from some very complex analysis in the most simple way is this common signature. So this cluster of good bugs, this cluster of bad bugs that's associated with good diet or bad diet and vice versa with good health outcomes. And I think something that we haven't really touched on yet is that how can we actually use these results for some sort of actionable outcome? And I think Tim will delve a little bit into this. But as a nutritionist, what I find really exciting is where we see the segregation of foods that might be we would consider to be quite similar. So where we have white bread, brown bread, the brown bread is associated with all of these harmful bugs, yet these favorable bugs, yet the white bread with these unfavorable bugs. And I think this is really important because we need to think about how we consume food in the broader context that, it's very much what determines our food intake isn't just our knowledge on what we're showing here, the health outcomes. It's around cultural, social, and kind of societal impacts. And so the findings that we've got here, where we have this segregation of bugs and this clear segregation of foods, enables us to still eat within these kind of constraints that many of us need to consume foods whilst making, healthy choices. So if we want to eat chocolate, fine, eat chocolate, but go for the dark chocolate. If you want to have bread, but go for the brown bread rather than the white bread. So there's some real simple, actionable outcomes that we can actually achieve from this, from what's actually very complex association data.
Great, thanks, Sarah. And so Tim, we've actually had loads of questions on this, and I think this is where kind of Tim is going to answer them. So, how can I eat for a healthy gut? How can I discover what bugs live in my gut? And how can I get involved in this research? Would you mind kind of just wrapping things up by answering those questions, I guess? Oops.
Sorry, I was on mute.
Okay.
So I think we've established there are some general principles anyway that all of us can get to a certain level of gut health. And I discussed these in some of my books, such as eating a diverse range of plants, and this the so-called trying to achieve 30 different plants a week, which is associated with diversity. But that only gets you so far. And this new study has told us that as we have a unique microbiome, there isn't a one-size-fits-all diet that's going to work for everybody. And this means that some people are going to do better on a high-fat diet, some people are going to do better on a high-carb diet. And so there are also things about timing of your foods, and this increased personalization really about how do we take what microbes you've got and actually try and move you towards a healthier microbiome state that's going to also help your sugar peaks, your fat peaks, etc. And this is really where we're going now. And the exciting thing is that we've done this 3 or 4 years of research, and now there is a commercial product available in the US that has been going now for a couple of months on a fairly small scale and is building up fast, so that we're combining data from your microbiome measured in the same way we've done in these studies with these metagenomes to give you an idea of what the bugs are inside you, combined with the measurements of your blood glucose and your fat levels through standardized meals, etc. And so from this, you can discover your own mix of good and bad microbes and understand how your own body responds to food. And this is very much the idea behind the Zoe program. But the good news for us scientists is that all the data that is being generated by this commercial project is going back into the research. So we can actually, every few months, unpick more about food that's crucial to everybody. So at the moment, if you like, we've found some fantastic insights, but there's so much more we can discover. And once we move from doing just thousands of people to hundreds of thousands of people, it's an extremely exciting time. So if you're in the US, you can get on board with this now and help us with our research by going to the website. If you're not in the US and say, for example, in the UK, we are doing our best to bring this to the UK this year. COVID has slowed us down a bit, but we're doing what we can, and hopefully within a few months, it will be available. So that's really it. But it's all about combining all these complexities into one, and you will need an app to get through this. And I think this is the new age, really, of the app. Days when you could just have a little card to memorize what foods to have and not have are long gone. We need sophisticated apps to take all this data together and using artificial intelligence, and that's really the exciting future.
Great, thanks, Tim. So yeah, so if you're in the US and you're watching this and you're thinking, wow, I want to know exactly what good and bad bugs are living in my gut and eat in a way that is really personalized to me, head over to joinzoe.com, and all the information you need to buy the test kit and program is there. And if you're in the UK or elsewhere, make sure to sign up for our mailing list if you haven't already, and we'll be in touch kind of when more information is available. So we've actually I was worried we'd actually go over time, but we've got a bit of time for some questions now. So I'm going to kind of ask a few as we go through. I'll direct them at, I'm not going to direct them at any specific scientists. So if you know the answer, please do pop in and answer it. So we've had a few questions about on the subject of food quality. Did we look into organic versus non-organic foods?
The answer is no, because it's very difficult to do at that scale. So there have only been a few studies really showing differences in organic versus non-organic foods that have produced significant results, because it's hard to get exactly like for like and balance all the other parts of the meal up. So often the person who's eating organic foods is going to be spending more on their food budgets, eat healthier than people in eating non-organic foods, and that's very hard to balance up. So that's again something we'll probably need hundreds of thousands of people to do properly. There is some data showing people who have microbes that are healthier, not our study, but others, or less healthy that are eating highly processed foods with lots of pesticides in them. But that's really much for the future.
Great, thank you. So I think there's a question here. So either, I guess, Tim or Nicola, I feel like this is for one of you. Is coffee good or bad for the gut?
Oh, absolutely good. Unless you've got some real problem. But and I said, every time we say good, it means for most people, because we're all unique. And some people do react badly to coffee, but all the epidemiology suggests that actually coffee is very good for your heart on average. So and we haven't really understood why. So now I think we've taken the epidemiology data, which shows that you have a reduced risk of heart attacks and heart problems if you're a regular coffee drinker, and now showing it's probably because of its effect on the gut microbes, probably because of the healthy chemicals in the coffee bean and the complexities when you ferment the coffee bean, you get hundreds of these defense chemicals, these polyphenols. So if you're a coffee drinker, carry on. And a few questions about caffeine. It does seem to be in the epidemiology that decaffeinated coffee seems to work nearly as well. So it may not be the caffeine that's crucial here. It's hundreds of other chemicals that don't get mentioned.
And I can add that a link between the specific microbes and coffee is kind of quantitative. So we see on the amount of coffee one drinks that the association is stronger or lower. Of course, yes, coffee, I think all epidemiologists are in agreement that that is good, probably at the normal levels, of course.
Good Italian coffee rather than American coffee, is that right, Nicola?
I would say so, yes.
we have a lot of relieved viewers that knowing that coffee is not terrible for them. Thank you. So I'm just trying to find a few because we've got a lot of repeat questions here. So how is it possible to estimate how quickly the microbiome can improve?
Yes, I think we have to estimate that. I think maybe Sarah can talk about the study another study that we have ongoing, maybe a bit of a spoiler.
You mean the Blue Poo study?
Yeah.
So I'm not quite sure how that specifically relates to this, but something that we've been looking at as part of the protocol from this PREDICT study is we feed participants these really delicious muffins that have dyed with blue dye, and we can measure something called transit time. So we can measure the amount of time it takes for the blue dye to go from your mouth to come out in your poo. And then based on the transit time that we can see, it gives us a really good idea of the diversity of your microbiome, but also the relative balance of some of these good and bad bugs. So if that question's in relation to how quickly can we get a rough guesstimate of your microbiome, you can go out and buy some blue dye, add it to a cake and time how long. As long as it's somewhere between half a day and about 2.5 days, then you're in that kind of happy place with some happy bugs based on what we've seen. If the question is more related to how long does it take, having changed your diet, to how long it changes your microbiome, I think this is something that Tim maybe has more of an insight than I do, but in brief, it can change very quickly in anything as short as he said earlier, a couple of hours to 3 days, you start to see quite big changes. Tim, do you want to add to that?
Yeah, it sort of depends. I mean, we know that for example, if you've never drunk coffee before and you suddenly start after this webinar drinking coffee, probably, you know, within 12 hours, your microbes will change. And so the question partly, it depends really on how extreme, the dietary change is. And like if you're a vegetarian, you suddenly start eating meat or vice versa, you can have an effect in a matter of days. If you're just subtly trying to change an already healthy diet to something a bit better, you might see changes in the microbes, but it could take you several weeks before you could actually notice it statistically. So I think it kind of all depends, but at a very individual level, your microbes will change pretty quickly. Now, whether a standard commercial test would pick it up is more difficult. And generally, most tests of dietary changes are done, you know, with a minimum of about a month to see anything, 1 to 3 months. And that's because you know, the tools aren't as precise in terms of picking that up. And because there's a lot of variability day to day between people and between individuals. So that's why it's a slightly tricky question. But if you may well feel better after a few days, if you might, you know, and you'll know that because your bowel habit will change. And you also know, you know, when you eat junk food as well, go the other way, it can go, you can feel quite bad within a few days as well.
Great. So we have time for one more question, I think. So do we know anything about how physical activity impacts the gut microbiome? And, you know, are there any other factors we looked into in the PREDICT studies to understand this link?
Nicola can probably answer, but I mean, in general, lots of people have tried to look at physical activity in the microbiome, and the effects have been always much smaller than diet in general, so it's been harder to pick up. There have been suggestions of a beneficial effect, and certainly we know that there's a link between exercise and your sugar peaks. And people vary a lot in how that works. Nicola, do you want to add to that?
I think yes, the perfect answer. Is not something we specifically looked into this study, but previous work said exactly what the team mentioned. Yeah.
Great. So we've had, I think, far more questions asked than we can answer. So I'm sorry if we didn't get to your questions. Thank you to everyone who's joined us today. I know this was a lot to go through, but it's really exciting. And I think what's so exciting is, you know, you really can take control of your health and weight with more personalized advice, food advice based on your body and your gut. So for those in the US, head over to joinzoe.com. For those not in the US, keep an eye out for our test kit and program launching where you are. And thank you, Tim, Sarah, and Nicola for joining me today. I had a really good time chatting to you.
Thanks, everybody.
Thank you very much.